it's called programmable recognition. So usually it should be able to, you can design it, you can design an algorithm to make a sequence easily recognize a defective gene. All right. So that defective gene is usually causing the disease. You will be trying to make it programmable and using some artificial intelligence to uh, go and find each defective genes and correct it possible. And we will show you example that it actually works. It can recognize defects and it can correct that gene. The cool thing about this structure, it's a multivalency, meaning there is not only one copy of the DNA, but hundreds of them. So all of them will work collectively to kill a disease causing gene. And we will show that it can recognize, when you program it, it can recognize a defective gene with really high precision because of the cooperative binding, again because of the multivalency. So just to quickly compare this new structure to what we know, linear DNA, this structure is similar to this when it comes to sequence. But when it comes to dimensionality, you go from one-dimensional to three-dimensional structure. That's all you do. You convert linear DNA into a 3D structure. And when you do that, you can bind tightly to defective genes 100 times stronger, because you're just moving from 1D to 3D. And it has some organic core, the gold. So gold, we can see it. So the drug is actually can be followed and can be seen because it has a core, gold as a core. All right, so good for diagnostic and monitoring with some artificial intelligence hooked up sensors. Now, the beauty of this versus this, it usually go inside cells because that's where our genomic DNA, they will go inside cells and correct the gene defect. Linear DNA cannot. If you throw linear DNA on a tissue or on a cell, it will never go inside. And that's the big advantage here. Another advantage is it can hide from our immune system. So it's really stable, as opposed to this, it will be killed quickly by our immune system. Now, I'm just gonna use one cancer type as an example, so we all understand the platform. This is prostate cancer, abbreviated as PCA. Now, with the prostate cancer is very, it's considered the second leading cause of cancer in men. And if you can detect this cancer and treat early before it becomes metastasized, then patient will have 100% chance to survive. But if it start to metastasize, then the patient chance to survive drop down to 28%. And this is current, this is today. Why? Because we cannot use the data that we have in a very intelligent way to correct that mutation, to detect that mutation, and so on. So we are still lagging behind when it comes to AI. So here's the goal. We want to use AI to not only detect, but also to cure. What I'm gonna show you is only detection, and what I'm gonna leave you with is your imagination to cure cancer. So one genetic material called microRNA. It's one type of RNA. We all know now about mRNA as vaccines for COVID. So it's similar principle. mRNA can fight against viruses like coronavirus, but it also can fight diseases like cancer. So if we can detect this microRNA and use it to kill cancer, just like mRNA vaccine, then we will have a really high chance to treat so many people. Because what I want to move on later on is from one health or one treatment for all or precision medicine. Probably heard about precision medicine in another workshop. So this genetic material is present in our blood. So if the cancer is in the prostate, it will release this genetic material in the blood, and you will see it in the blood. So easily can be detected. The problem is it's very short, so we will have to, and very little. So the concentration wise is very little. So we will have to use nano to see it. It cannot be seen by any machine we know, unless we do some modifications and sophisticated uh, um, techniques. But with nano, we can see it, and we can hopefully treat it, as you will see later on. So that's the biological marker, similar to DNA, more similar to RNA, but it's called microRNA. That's the focus of the next slide. So here's what we did. We used that spherical nucleic acid that I just described, and it's programmable. It will uh, detect microRNA number one, let's say, that is specific to prostate cancer, and you can then program it to detect gene specific to neurological disease or diabetes or you name it, any disease, or maybe coronavirus. But you need a lot. You need huge data. We have thousands of them, and you can create hundreds of thousands of programmable spherical nucleic acids. Basically, you can use AI to digitalize drugs. It's not pre-made drug. We will listen to programmable or AI to design the drug for patient one, and then listen to AI to design drug for patient number two, because the same drug does not work on two patients per se. So each spot is one genetic defect, and if it's lighting really shiny, this means that copy number is really high. And to treat a patient, we need to correct all of those copies. That's maybe million copies of gene defect. If it's dim, then the copy number of that defective gene is low, so probably easier to treat. And you can see in one chip, you can profile whole population. For example, maybe this signature is specific to prostate cancer, but then this signature is specific to another disease. And then you can literally profile hundreds of diseases, hundreds of patients in one shot. So what they did is they actually applied this nano technology, and they found it can detect and recognize a defective gene, for example, MER16. And if that gene is different by one mutation, one gene mutation, they call it SNP, okay, single nucleotide polymorphism, then it will not see it. So this is how the beauty of AI here is, that we can make nanotechnology to see genes, defective genes, even if they have one single nucleotide polymorphism, while wild type is not touched. So if your gene, what they call it wild type, and wild type means healthy gene. If the healthy gene is present, those nanomaterials should not touch it. But if the healthy gene become diseased and carry a mutation, then it should be able to recognize it with your programmable algorithms. The next slide. So it was applied on so many patients, maybe hundreds of patients with the prostate cancer, and found a really nice pattern. 5 genes, 5 microRNA are enough to tell me if the patients are healthy or if the patient has low-grade cancer or if the patient has high-grade cancer. And if you notice here, those patients are misdiagnosed. Physicians thought they are low-grade cancer, but it turned out they are high-grade cancer, meaning they will die because of cancer if we cannot treat them. Okay? So 3 distinct subgroups. Those microRNAs, sometimes they themselves carry the gene defect. So if they go high in number, they lead to cancer. And if they go low in numbers, in other cases, they also lead in cancer. The difference here is that we call them here tumor suppressor, like soldiers. So if we don't have cancer will develop. And we call them here oncogenes, meaning they are causing cancer. So if we have a lot of them, then cancer will be developed. So I'm gonna just show you an example of technology, how to treat, and then we think about programmable digitalized drugs for precision medicine. So let's use the same technology, spherical nucleic acids. And we know they go inside cells. I will show you proof in the next slide. When they go inside, they bind to the gene causing cancer. And then when they bind it, basically you can kill cancer. If the gene is not present, they will not touch other genes. Other genes are protected. Only genes with mutations will be recognized, and then cancers will die. So this process is programmable. This process is digital. That's what I want to emphasize here. So I promise to show you that these spherical nucleic acids, we call them SNAs, just an abbreviation. They go inside cells. Look, in half an hour, they're already inside cells. Those are cancer cells. After 2 hours, they are really inside cells. Well, how do we know that? Because they are shiny. They have gold. You can label them with colors, and then they will make cells shiny if they are cancerous. The same DNA, one-dimensional, throw them on cells. They will never, ever be able to go inside cells, even if you waited for a couple days. As evidence here, all of those are not shiny, meaning this cannot go inside cells and cannot programmably recognize the defective gene. SNAs, they can easily go inside cells. So this is really versatile, powerful technology, ready to be digitalized, ready to be programmable, to treat not only one disease, but so many diseases. And that's the beauty. You change the DNA sequence, you change the drug, as easy as that. So this is just for example. In prostate cancer, there is a defective gene called MER21. The gene is known to regulate so many important genes like PTEN. All of those consider them as tumor suppressors, meaning soldiers. They protect us from cancer. This guy is actually down-regulating the tumor suppressors, meaning it makes less, our cells makes less copies of those. When this happens, you can see cancer proliferate in high rate and divide really quick and fast, and then it can metastasize. And that's why we classify it as oncogene, a gene that causes cancer. All right. So here is the beauty of this technology. We get the sequence of that gene. It's really simple, ATCAACAT, well known, and that's the length you will work with to program a drug for any given disease. Okay, so when you design your DNA to be like this, it will go after and recognize that defective gene. And when it recognizes it, will basically kill it and kill cancer cells. Look at this. Here, patient with cancer can metastasize and fill the whole area. It was empty, like just think of it as another organ. They can move from one place to another, metastasis or cancer spreading in 2 days. In 2 days, the spot is already filled. With that SNA, you can slow down cancer metastasis. If you want, you can make the programmable even better to kill cancer as well. But it's obvious that cancer progression is slowed down as evidence from here. Now, if we go next, we will observe really interesting phenomena. Same SNA can kill one cancer and then get out and then go to another cancer cell and it can kill it. Why? Because we see it outside cells. So it'll do its job in cell number one and then leave and attack another cells. And called lipid exosomes. They are inside lipid exosomes, as clear from here. The what does that tell us? It tells us that they become smart, they become intelligent. They go from one cell to another, hiding on those exosomes, knowing where cancer is. Because they left cancer, now they have the same recognition element as cancer cells. So the second cancer cell will be fooled, will think this is self. And so it will allow it to go inside, infect another cancer cell, and regulate that cancer cell, slow it down, and perhaps kill it, right? Because now we started with the stupid SNA, and then we ended up intelligent SNA. We can use less concentration of that drug. Look at this. We started with 1,000, and when it comes to concentration, and now because they become really intelligent, we only need 0.35 as opposed to 1,000. That's 3,000 fold less drug can be used when we apply artificial intelligence to get the same result, to get the same cancer and probably cancer killing. All right. So what I hope this is clear, because now I'm moving to a more broader area. The broader area here when you do personalized or precision healthcare, when you invest in artificial intelligence to digitalize the pharma industry and to digitalize drug industry, so each patient will have a different drug. I wish I can convince you that very good market. All of these companies are employing artificial intelligence to achieve precision healthcare, and all of them actually making billions of Saudi riyals. For example, in 2008, the market was 300 billion Saudi riyal. It's expected to jump in 2028 to 800 billion Saudi riyal. That's a big jump, 10% increase. Okay. The another encouraging things is when you apply programming to digitalize the drugs, then you uh, follow the step of those companies. As it says here, already tens of drugs approved by the US FDA and possibly by the rest of the world. And here are some just examples. Inherited diseases like, brain diseases like autism and so on, cancer like lung and breast. So still a new field. We don't have much drugs that we call programmable drugs or digitalized drugs that uses AI. Um, the second problem why this is good for our country, Saudi Arabia, is because we spend a lot of money on, because we have what we call intermarriages. So cousins can marry each other. When cousins, relative to each other, you increase the chance of genetic diseases. It's obvious everywhere. Saudi Arabia and Pakistan, the only two countries that have high intermarriages. And because of that, we spend 6 billion Saudi riyal annually to treat diseases that could be prevented with AI. And remember, when you correct a gene, hopefully coded for good, not for that patient, but also for his or her offspring. Okay, like children, generation one, generation two, and so on. So this is a very good way to permit intermarriages, but with AI we can do some maintenance and remove those genes that are causing diseases. So everyone has the freedom to marry his cousin or her cousin and so on. The last slide, and I will conclude. And actually, two more slides. So because of that, it's like England, UK, Finland, and Iceland, and so on. They started to seek. All right. More than 100,000 patients were sequenced. Their whole genome is sequenced. And those include patients with cancer, around 40,000 patients, and also rare diseases, around 90,000 with from patients with rare diseases. So they sequence. We have so much data and we have so many mutations that are linked to diseases. You can see here this is how many genomes were sequenced, and this is the result analysis. It's around 30,000 mutations for cancers and around 70,000 for rare diseases. Now we know the mutation, okay? And that's what I want you to focus on. What are we doing right now? Just diagnosis. We just tell people you have the mutation, don't marry your cousin. That's all we can do. And I intend to explain programmable SNAs so you can think of going beyond diagnosis, because diagnosis already done in so many countries, including Saudi Arabia, as I will explain in the next slide. But no one dared to use artificial intelligence to design programmable therapies for hundreds of thousands of people. Saudi Arabia did its share. We sequenced 50,000 Saudis, so we have the genome of 50,000 individuals. And you can imagine how many diseases we know now. But trying to, because no one is able to adopt artificial intelligence with the programmable gene therapy. Okay, so that's what I want to leave you with. Hopefully this will help you think and think and think and propose something impactful. I'm sure you guys are the future inventions, and I'm sure your thoughts will matter. So I want to thank everyone. If you have questions, you have my email here. You can email me with the questions and help. And also, I don't want to forget to thank the organizers again. Jama did a wonderful job organizing this event, where شكرا لهم على الدعوة الكريمة. هاكاثون مكان للإبداع والابتكار، وبرضو الأندية المنظمة نشكرهم والمجهود الرائع. الجميع شكرا للحضور، شكرا لاستماعكم، وأتمنى أن أفدتكم ولو بشيء القليل، وأعتذر إن كان في أي تقصير. شكرا لكم.
الشكر والتقدير لكم دكتور، وأقدم اعتذاري على الخطأ في اسمكم الكريم. معكم دكتور علي الحسن.
ولا يهمك، شكرا لك.
العفو. دكتور، تبغى تفضلوا بالأسئلة ولا يتواصلوا معك على الإيميل؟
اللي تحبوا. أسئلة I'm open، يتفضلوا، وإذا يبغوا وقت يفكروا ويرسلوا بيل برضو حاضر. الاثنين مناسب.
يلا مشتركين، هذه فرصتكم للاستفادة من علم وخبرة الدكتور علي. تفضل. في أحد حابب يشارك؟ للآن دكتور ما في أي مشاركات. كل الشكر والتقدير لكم دكتور، كانت ورشة ثرية وممتعة، إن شاء الله يكونوا المشاركين استفادوا منها أكبر قدر من الاستفادة. شكرا لجهدكم ووقتكم.
إن شاء الله، إن شاء الله يا رب. شكرا لك أختي رغد، وشكرا للحضور جميعا.
العفو دكتور، يعطيكم العافية.
مع السلامة.